So hello students and welcome back again to another video tutorial by Genome Biotech. In this video tutorial, we would be talking about the holiday model. So what is the holiday model? How holiday model is being used to explain the process of homologous recombination that we must know. We must try to understand in what manner the holiday junction takes place, how the holiday model is being proposed in order to form the holiday junction. What is this holiday junction? How does the formation, progression, and the resolution of holiday junction is going to take place? First, we must know why this holiday model is important and what is the need of this holiday model. The holiday model is just a simple model which is useful for understanding the steps of homologous recombination. Okay, or I should rather write something like this: a holiday model, a holiday model is a simple model to explain the process of homologous recombination that occurs during the process of crossing over, during the process of crossing over at the time of gamete formation, at the time of gamete formation. Okay, so this is a process that is going to generate the variations. Okay, so it is the main process responsible for the generation of continuous variations. So all the continuous variations are being produced as a responsibility of the process of homologous recombination, which is also referred to as the model behind the process of crossing over. Now, how does crossing over take place? What is crossing over? Crossing over refers to the exchange of chromosomal segments in between non-sister chromatids of the homologous chromosome. So if you look here, just a small diagram to show it to you all. In this diagram, if I do make certain chromosomes, if this is one chromosome, okay, this chromosome has made two different arms. Okay, this is one arm, this is another arm. This is the chromosome number one copy, and this has been inherited from the paternal, that is from the father. And there is another chromosome that is present, and that chromosome is being inherited from the mother side. Okay, that is the maternal copy of the chromosome. Then you know that these paternal and maternal copy which is being inherited in the person can undergoes crossing over. What is crossing over? Crossing over refer to the exchange of chromosomal fragment. Now these are the two chromosomes. There is one chromosome and another one chromosome. These two chromosomes can undergo the crossing over at their junctions. Okay, so they can undergo crossing over. These can undergo several different types of crossing overs. Now, what will happen in the case of crossing over? They will exchange their chromosomal fragment. Their chromosomal fragments will be exchanged. The black one will go to here in the white one, and the white one will go to this one. Then what will happen after the process of crossing over? There would be new variations that would be produced. It would be something like this. Okay, it would be something like this: a part of the chromosome would have been exchanged. Okay, and plus this part of the chromosome would have been made. It would be like this: the entire chromosome being black, being made black, except this part. So this is the part that has undergone crossing over. The chromosomal fragment that had undergone crossing over, that had undergone crossing over. Okay, this is the part that has undergone crossing over, that has been crossed. Okay, so now how does this process of crossing over take place? Now what will happen after this particular incident? The gametes will be produced. These will be the four gametes that would be produced: first, second, third, and fourth being produced. Here the chromosomal fragments have been exchanged. This is the chromosomal fragments that have been exchanged. You can look here. Okay, this is the chromosomal fragment that has been exchanged. Here you can find this is the part of the chromosome that has been exchanged. So here you find that out of the four one chromosome gametes, none of them are resembling the parents. The parents were of the following types. One was like this, another one was like this. See, 50% is parental type. This is parental. This is parental. These two are both recombinant. Recombinant. How these recombinants are being produced? These recombinants are being produced as a result of crossing over. Now, the process of crossing over takes place with a model that is referred to as, it is being explained with a model that is referred to as the holiday model. So without wasting any time, let us move forward with the holiday model and try to explain how does holiday model work. Okay. In the meantime, if you feel any question, just message me. Next point.
While it is now clear that most recombination events involve synthesis of new DNA, a feature absent from this model, the Holiday model is still useful for understanding recombination.
So holiday model is going to explain us which are the important features which are being required for the process of recombination to take place.
When you have completed this exercise, you should understand how strand invasion can create a Holiday junction, understand how branch migration leads to recombination, and understand the differences between the two possible modes of Holiday junction resolution.
So the aim of the exercise is to understand the process of strand invasion. How does strand invasion take place? What is branch migration? How the DNA branches are being migrated, and how the holiday junction is being resolved in order to form the recombinant or the hybrid DNA molecule. How the new variations are being produced by the resolution of holiday junction. Kindly look forward.
To visualize homologous recombination using the Holiday model, it is useful to first picture two homologous double-stranded DNA molecules aligned with one another. These DNA duplexes may carry different alleles of the same gene. In the first step of the Holiday model, single-strand nicks form at identical positions in each DNA duplex.
So now, looking at the DNA duplex, you can find if you are imagining a double-stranded DNA molecule having the same homologous chromosome. Homologous chromosome means if you are talking about the first homologous chromosome, if you're talking about the first chromosome, this is also the first chromosome that is present. Both the homologous chromosomes will cleave the DNA at a particular site. Okay. Both the homologous chromosomes will have a cleavage at a particular site. Now, supposingly there is a particular site cleavage that is going to take place here. There is another cleavage that is going to take place at this particular site. So the two strands of the DNA are being cleaved alternatively with regard to each other. Now the exchange of the genetic material is going to take place. How now the branch migration is going to take place. Now how branch migration is going to take place, that we have to see. Please look forward.
DNA strands near the nick sites peel away from their complementary strands and base pair with the homologous duplex. This step is called strand invasion. The DNA intermediate in which two DNA duplexes are connected by crossing DNA strands is called a Holiday junction.
Please look here. See, once the strand cleavage has taken place, then after the nicks have been produced, the strand began to migrate into opposite strand. This step is called as strand invasion. The step of homologous recombination in which the initial short region of the base pairing forms between the two recombinant DNA duplexes. There is a region of base pairing which is being produced, and the initial region of the base pairing formed between the two recombinant DNA duplex. There is a region that is being produced, and this region is being formed in between the two homologous chromosomes having a branch point, and this branch point will be referred to as the holiday junction. Holiday junction is a DNA intermediate in which two DNA duplexes are connected by a crossing DNA strand. There are two DNA duplexes, and both the DNA duplexes are being arranged in the form of a crossing DNA strand. So they are present in the form of a crossing DNA strand that you can visualize here. This is the cross that is being going to be produced. Next.
Remember, Since the duplexes are identical, or nearly identical in sequence, each of the nicked strands is able to base pair with the uncleaved strand on the opposite duplex.
Now see, as the duplex are completely complementary to each other, so each of the strand that is migrating will be able to form a base pair with the pre-existing strand of the DNA molecule. So every strand that is base pairing will be migrating and base pairing with another strand of the DNA. But as the alternate forms of genes are present, for example, there is capital A, another one is small a. For example, there is capital B, that is the dominant gene is present. Another one is a small b, that is the recessive gene, that is going to migrate. And when the migration is going to take place, then the complementary bases will form a hydrogen bonding with regard to each other. Next.
The holiday junction can move along the DNA by repeated melting and formation of base pairs. Each time the junction moves, base pairs are broken in the parental DNA molecules, and identical base pairs are formed in the recombination intermediate. This process is called branch migration.
Now, the third point takes place. One is referred to as endonuclease nicking. We have seen endonuclease nicking. Then the base pairing was taking place. And last is referred to as branch migration. Now, how does branch migration takes place? The old DNA strands are being broken and new are being formed. Then this process is referred to as branch migration. You can look here. There is a melting of the base pair at the front of the replication fork, and there is a formation of base pair at its back. This process is commonly referred to as branch migration, the step of homologous recombination in which the holiday junction moves, increasing the length of the exchange DNA molecule. There is a length of the exchange DNA molecule that is being enhanced or increased. This process is commonly called as branch migration.
Different alleles of the same gene carry small sequence differences. During branch migration through these regions of sequence difference, DNA duplexes are formed carrying one or a few sequence mismatches. These regions are called heteroduplex DNA.
Okay. Now there are regions which are being produced, which are referred to as heteroduplex DNA. What do you mean by heteroduplex DNA? Heteroduplex DNA refers to the region where there is a mismatch which is present in between the two DNA strands as the DNA has migrated. So this DNA being carrying a different allele, different form of the allele. For example, there was capital B, here it was a small b. So this small b, when it has migrated in front of the capital B, of course, there would be regions where mismatch will be present. There would be regions where there would be no complementary base pairing or no effective matching taking place. Okay, so these are the regions where new variations will be introduced. These are the sites where new variations will be introduced. Now, how these variations are being introduced, that we have to see.
The final step of DNA recombination is cleavage of the holiday junction to regenerate two separate DNA duplexes. This process is called resolution. To aid in the visualization of this step, the holiday junction is rotated to give a square planar structure with no crossing strands.
Now, this is very, very important. Now, see, once the holiday junction, the DNA has been migrated, the holiday cross junction is being produced. Cross junction refers to a junction in which there is a formation of a cross like structure. Now the resolution of this holiday junction has to take place so that the DNA can be effectively resolved. Please again look here.
The final step of DNA recombination is cleavage of the holiday junction to
Now the final step would be the cleavage of the holiday junction, regenerating the two DNA duplexes. How the cleavage is going to take place? You take this DNA molecule and try to rotate this DNA molecule via 180-degree. Look here.
generate two separate DNA duplexes. This process is called resolution.
See
To
this.
aid in the visualization
This process of rotation is called as resolution.
of this step, the holiday junction is rotated to give a square planar structure with no crossing strands.
Now there are two different sites where from this holiday junction could be effectively cleaved. Those two different sites are either in the form of a patch cut or in the form of a splice cut. Okay, it could be top to bottom cut. Okay, or it could be right to left cut. Okay, patch and a splice recombination products could be produced. This is the way by which the resolution of the holiday junction can take place. Now, how it is going to resolve, let us have a view.
Resolution can occur at one of two possible sites.
Okay,
Cleavage
at
at
the
each
two
of these
different
sites
sites.
results in a different genetic outcome.
Now, which are the two different sites and how the cleavage is going to take place at the first site and the second site? Which are the different products which would be produced? Now, if you look at the first site, if cleavage is going to take place at the first site, do you know what would be the result produced? Capital A, capital B allele along with small c. Capital A, capital B, then the third one would be small c. And here it would be produced will be capital A dash small b dash, and here after the cleavage at the green site is taking place, then a small c will be produced, a small c and a small c. And in the other types of cleavage will take place, then capital A small b dash and a small c dash will be produced, and it would be A, B and C that will be produced. Okay, look here.
One of the possible cut sites is in the pair of strands that remained uncut during the first step of recombination. These strands can be easily identified because they each consist of a single color at this point in the model.
Okay, now how the cut is going to take place in the first step. In the first step, the cleavage is going to take place in this manner so that the two strands can be resolved.
The resulting duplexes are called splice recombination products
This is a
because the two
splice.
original duplexes are now spliced together, with the DNA sequences flanking the site of recombination swapped. This may not be obvious in the short stretches of DNA shown, but should become clearer in the context of longer DNA duplexes.
Now, the questions that will be asked to you: where is the patch recombination product? Where is the splice recombination product? Patch recombination product always involves cleavage at a particular plane. When cleavage is at in plane, when cleavage occurs in plane, when cleavage occurs in plane, occurs in plane, in plane, then it is referred to as, when cleavage is going to take place in plane, then it is referred to as patch recombination product. Patch recombination product. Recombination product. Combination product. This is a patch recombination product that is going to be produced. Okay, so one is referred to as the patch recombination product that is going to be produced when the cleavage is occurring in a particular plane. When cleavage occurs in a plane, then the recombination product which is being produced is referred to as the patch recombination product. When the cleavage is going to occur at 90-degree, then it will lead to the formation of the splice recombination product. When cleavage occurs at 90-degree, then it would be referred to as splice recombination product. When cleavage occurs, when cleavage occurs at 90-degree, occurs at an angle of 90-degree, at an angle of 90-degree with respect to the plane, with respect to the cleavage is referred to as, the cleavage is called, the cleavage is called splice. The cleavage is called is called splice recombination product. Splice recombination product. Combination product. This would be referred to as splice recombination product. Okay, this would be produced. Any doubt? Any problem? The product that is being produced is a splice recombination product. Moving forward,
The resulting duplexes now carry one pair of alleles from one parent, a region of heteroduplex DNA, and the pair of alleles from the other parent. Since the C alleles have switched from one duplex to the other, these products are also called crossover products.
Now, see, when the splice recombination product has taken place, this is splice recombination product, then you get identical set of genes in the A allele. You get heteroduplex. Why? What is heteroduplex? Heteroduplex means that out of capital B and B dash, now what you have is capital B and small b dash. So this fragment has been exchanged. The chromosomal fragments have been exchanged. That is why you're getting a capital B and a small b dash. So this is referred to as heteroduplex. And the third one would be referred to as the crossover products. In the case of crossover products, what is going to happen? There is going to be a dominant gene present in one, recessive in another. Now the crossover products have been produced. Now the one which was having the dominant will have the recessive one. You look here, this one was the parental. Now look at my cursor. This one was the parental. Now this has been replaced with the allele, small allele, that is small c and small c. So this becomes the crossover hotspots. Okay, and these are the crossovers which are being produced. Such type of product which is being produced is referred to as the splice recombination product. This is called as the splice recombination product. This is splice recombination product that is going to be produced. ये splice recombination product है जो कि यहाँ पर produced हो जाता है. Any doubt you have, you can just ask me. This is splice recombination product. Okay, sorry. Okay, this is the result that you would be getting. Now the next type of products that would be produced. Okay, crossover outcomes, heteroduplex and identical. Such type of questions will be asked. You look at the questions also.
The
Next.
second possible cut site occurs in the pair of strands that were nicked during the first step of recombination.
Now
These
this,
strands can be easily identified because they each consist of a mix of colors at this point in the model.
please look here. The cleavage can also occur at the second site. This second site is planar because the cleavage is occurring at the plane. So that is why it is referred to as the patch recombination product. Cleavage occurs at a planar site, at a planar site. So, so such a cleavage is referred to as, so it is called as, so it is called as patch recombination product. This is the patch recombination product that would be produced. Okay, so this would be referred to as the patch recombination product. You can look here. This is the patch recombination product which is being formed when the cleavage occurs in a planar site. Is this clear? Moving forward.
The resulting duplexes are called patch recombination products because they contain a patch of hybrid DNA. Since the A and C alleles are still present together on one of the product DNA duplexes, these products are also called non-crossover products.
Now, why they are referred to as patch resolution product? Because the central DNA molecule is actually present in the form of a particular patch. You look here. This is the DNA molecule that is present in the form of a patch. Just have to look here. There is capital A, capital B, capital C, capital A, capital B dash, and capital C dash. What are the products that you are obtaining? Capital B and along with that small b dash. Okay, so this is a patch. Capital B dash along with a small b. This is a patch. So the patch is being created at a particular site. You look here. I'm just marking the site of the patch. This is the site of the patch where it has been produced. This is the site of the patch where the patch has been produced, and that is why this is referred to as the patch recombination product, in which firstly the set of genes are identical, the second is having a patch, and the third one is again identical. So the set of genes could be identical, patched, and then identical. Next point.
How well do you understand the holiday model? In this section, you will find out.
Now the next question.
Question 1. Put the steps of the holiday model of homologous recombination in order. You may have to use a step more than once.
Now, how the steps should be kept. Firstly, a single strand nick forms on both the DNA duplexes. Okay, there is a single strand nick that is going to be produced. Base pair forms. Base pair forms between the two recombining DNA duplexes. There is a formation of the base pair. Once the base pair has been formed, the holiday junction is going to move. As the holiday junction moves, the exchange of the genetic material is going to take place. And lastly, the holiday junction is cleaved, regenerating two separated DNA duplex molecules. Now, look here. This one was the correct answer. Okay. Second point, base pair forms between the two recombining DNA duplexes. The holiday junction moves. The holiday junction is being cleaved, regenerating two separated DNA duplexes. Look here. This is the first cleavage site. This is the second cleavage site. The patch and the splice recombination products will be produced. The next question.
Question 2. Which site must be cleaved to form each pair of products shown? Drag the products to their matching cut sites.
Now, you know that when the cleavage site has to be present in the form of a particular plane, it would be referred to as a patch recombination product. What happens in the case of patch recombination product? In the case of patch recombination product, what is being done? This is a planar cleavage. This is a planar cleavage. Planar cleavage means patch recombination. Okay, patch recombination. In the case of patch recombination products, there is a patch that is going to be produced. In the case of patch, the centrally present DNA will be exchanged. Okay, now you look here. This is capital A, this is capital A, capital B, capital C should be there. But the central part as well as the third part has been exchanged. So this is actually a splice recombination product. Splice recombination product. Recombination product. And another one here that you can find is that the central portion has been exchanged. So this would be a splice recombination. This would be a patch recombination because only the patch of the DNA has been exchanged. Now only the center part of the DNA has been exchanged. That is why it would be referred to as a patch recombination product. You just have to look. This is the patch that has been exchanged. Okay, so take this and put it here. So let me just took this and put it in the patch and take the other one and put it in the splice.
Correct.
Okay, moving forward. See this? These are the two DNA strands. Just clearing all the annotations, cleared all the annotations. You look here. This is the splice recombination product. See this? The splice recombination product. What we found in the case of splice recombination product is that see this, in the case of splice recombination product, there is identical of parental, then there is the heteroduplex, and then there is the crossover products that have been obtained. In the patch, there is identical DNA and there is a heteroduplex DNA produced. This is how the DNA molecules are being resolved.
Question 3. Match the terms with the products they describe.
Match the terms in the products that these are being described. Second site which is present, the second site which is present would be referred to as the patch recombination product. Okay, so I'm just taking it and writing it patch recombination product. Okay, and drag the items to the correct boxes you may need. Okay, splice recombination product is this. Okay, splice always produce crossover products and patch always produce non-crossover products. Now what? Okay,
Correct.
so that's in.
Question 4. If the 3' 5' strand of each parental DNA duplex is nicked to form a holiday junction, Which strands of the parental DNA duplexes are cleaved during resolution to form the products shown?
Okay. Which of the in the 3 prime to 5 prime strands of each parental DNA duplex is being nicked to form a holiday junction? Which strand of the parental DNA duplex are cleaved during the resolution to form the products produced? Now, which click on the parental strand to select them? Okay. These are the parental strand and these are the offspring strands which are going to be produced. What we have to select is to form which strand of the parental DNA will be cleaved. Okay, which strand of the parental DNA will be cleaved? If one has to, then strand has to be cleaved. How? Because you will have to cleave the C strand so that the C strand could be exchanged with the blue strand with this strand with the with capital C strand. How can we obtain a capital C here? This is capital A, this is capital B, this is capital C. Do you get capital C there? Capital C is here. Okay. And then the other strand that we are going to get it is small c, is small b, and A. So these are the strands that are going to be cleaved. Okay.
In the first step of the holiday model,
Yes.
single-strand nicks form at identical positions in two homologous DNA duplexes. DNA strands near the nick sites peel away from their complementary strands and base pair with the homologous duplex. This step is called strand invasion. The crossing DNA strands linking the two duplexes form a holiday junction.
Okay, this is the holiday junction that is being produced. This is the last slide, just a
The
revision.
holiday junction then moves along the DNA by repeated melting and formation of base pairs. This process is called branch migration.
This process in which the migration is going to take place is referred to as the branch migration that is going to occur next.
The final step of DNA recombination is cleavage of the holiday junction to regenerate two separate DNA duplexes. This process is called resolution. Resolution can occur at either of two sites.
There are two sites where the resolution can occur, the patch and the splice recombination product. This is how the recombination takes place and the resolution occurs. Okay, splice recombination product, and the last would be the patch. Okay, second side. Please note, take a snapshot of this particular slide, the most important one. That's all over. The topic is over.
You have completed this exercise.
Okay, this is all about this particular exercise. This exercise is completed. Now, in our next exercise, what we have to do is we have to study about how the process of homologous recombination actually takes place, which are the enzymes which are being involved. So we will have to look at the RAC BCD pathway of the homologous recombination. Okay, so let us start with the RAC BCD pathway of the homologous recombination, and then learn about the holiday model as well as the homologous recombination, how both the processes are going to take place. Okay, so that's all. I will have to take a break of 5